Good morning, ma'am. My name is Tashu Singh, roll number 24 from group 7-1, and the topic for my presentation is computational biology. Firstly, what is computational biology? Computational biology encompasses all computational methods and theories applicable to molecular biology and areas of computer based techniques for solving biological problems. Computational biology includes proteins. They are the building blocks of a living organism. It is a large molecule that is composed of sequence of amino acids. There are 20 amino acids which are divided into classes: hydrophobic, hydrophilic and polar. Moving further, DNA. DNA is a DNA or deoxyribonucleic acid is a blueprint of living organism. Secondly, DNA is composed of two strands held by a weak hydrogen bond. Each strand is a sequence of nucleotides. DNA has four bases which are classified as two chemical types. The bases of DNA are adenine, thymine, cytosine, and guanine. Moving further to the next slide, here is the structure of DNA which is shown here. Then moving further to the next slide, it is RNA. RNA is chemically very similar to DNA. Secondly, there are two most important differences. First, it is four bases present in RNA are adenine, guanine, cytosine, and uracil. Secondly, RNA nucleotides contain a different sugar molecule which is ribose. Moving further to the next slide, it is genetics and evolution. So, mutation, natural selection, and genetic drift. Mutation, the changing of the structure of a gene resulting in a variant form that may be transmitted to a subsequent generations caused by the alteration of single base unit in DNA. Natural selection, the process whereby organisms better adapted to their environment tend to survive and produce more offspring. Genetic drift, variation is the relative frequency of different genotypes in a small population. Moving further to the next slide, finite state and machines in BLAST. Firstly, it is used to find out which of the sequence in a database are related to the given sequence using BLAST. The BLAST system is a three-step process. Moving further, it's HMM for sequence matching. HMM are used when sequence become fairly diverse. Secondly, we can capture the variations among the members of the family and probabilities associated with them. So, by using HMM, we can find the best alignment between two sequences from which family does a given sequence belong to. Moving further, here is the example of HMM describing protein sequence family. Then, moving further to the next slide, here is the RNA sequence matching and secondary structure prediction using the tools of the context free languages. In RNA, a change to a single nucleotide is a stem region could completely alter the molecule shape and its function. So, and change in the stem can must be matched by corresponding change in the paired nucleotide. Context free languages are used to describe these nested dependencies and secondary structure. Moving further, here is the example of it. Then, complexity of algorithm used in computational biology. Firstly, approaches to many of the problems described here are computational, like breaking up of large protein and DNA molecule into substrings. Then, secondly, it is NP. Then, conversion of decision problem. S is a set of string and there exists some superstring T such that every element of S is a substring of T and T has length less than or equal to K. Here is the end of my slides. So, thank you, ma'am.